Welcome to the Wall Exchange. My name is Brett Finlay. I'm a distinguished professor at the Peter Wall Institute at UBC and a professor of biochemistry, molecular biology, microbiology, immunology. That means I don't know what I'm talking about. I'd like to thank everyone for coming to this event tonight. The Wall Exchange is a series of downtown lectures hosted by the Peter Wall Institute for Advanced Studies at the University of British Columbia. The Institute is dedicated to advancing innovative and fundamental research that can generate new ideas, new thinking, and ultimately make important contributions to society. The Wall Exchange objective is to help us collectively engage in conversations about important and timely issues within our global society, and tonight's conversation promises exactly that. So first of all, I'd like to thank the members of my band. These are called the Straight Jackets. You can guess why. They're Jerry Bowie on trumpet, who's a cosmetic surgeon when he's not doing these kinds of things, Andy Stack on guitar, Mark Bender on bass, and Johnny Boogie Seguin on drums. And I dabbled on sax. Thanks, guys. I'd also like to thank the Georgia Straight Magazine and Atai for co-sponsoring this event, as well as CBC for lending us Kelly Crow, and a medical sciences reporter for CBC, National News, as well as moderator for this evening's question and answer. Also, CBC Radio's One Ideas program will tape this event and broadcast it later, and for a future Ideas broadcast. Also, CITR, the UBC student radio station, helped sponsor this. So the program this evening, Dr. Bonnie Bassler will speak for 45 minutes, and then Kelly Crow will then serve as a moderator for another 45 minutes of questions and answers. Kelly Crow joined the CBC National in 1991. During her career, she's reported on a wide range of stories, including elections, floods, forest fires, political leadership conventions, and breaking news such as the 1919 Columbine shootings in Colorado, and, dear to my heart, the SARS outbreak in Canada in 2003. I must admit, I've been bugging Kelly for years. We've got to do more about these microbes. She's heard us. She talked a lot about antimicrobial resistance, for example, and we've interacted a lot during the SARS crisis and, over the years, several different items of microbiology. So thank you, Kelly, for helping with this tonight. So hopefully behind me on the screen, you can find our Twitter handle, @WallInstitute, and the hashtag #WallX. In addition to the microphones at the front, we'll be taking questions tonight via the hashtags, #WallX. You can send your questions in during the lecture and in the first 2 minutes after the lecture finishes. Our moderator will ask a handful of them tonight's speaker during the Q&A period. I should also mention that tonight's lecture and Q&A will be made available as a video and podcast on our website, where there's many other excellent talks. Now, to introduce Bonnie Bassler. So Bonnie is an investigator with the Howard Hughes Medical Institute. This is an extremely prestigious medical institute in the States, and the Squibb Professor and Chair of the Department of Molecular Biology at Princeton University. The research in her lab focuses on chemical signaling mechanisms that bacteria use to communicate with each other, commonly called quorum sensing. Therapies to block quorum sensing activity may represent an important new strategy for combating bacterial infections. Her research has revealed new insights into the basic biology and ecology of how these bacteria work, and findings that have made direct application in the future treatment of diseases. So Dr. Bassler is a passionate advocate for the diversity in the sciences. She's actively involved in and committed to educating the public in science. She's a member of the National Academy of Sciences of the USA and the American Academy of Arts and Sciences, and was nominated by President Barack Obama to serve on a board for the National Science Board, a board that oversees National Science Foundation and prioritizes NASA's research and educational priorities in science, math, and engineering. On a personal note, I've known Bonnie for many years. She livens up any meeting we're at, as you'll see tonight. And unfortunately, often I end up talking after her, which, as you see tonight, is a really tough act to follow. So please join me in welcoming Dr. Bonnie Bassler. Thank you.
Thank you. Thank you, Brett, for the really lovely introduction. I am so delighted to be here. I'm having a wonderful week. I've never had a warm-up band before, so that's actually a new thrill for me. And so I just want to thank the Peter Wall Institute for inviting me here and CBC for making this really interactive evening. And I want to thank you already for coming. And so let me jump into what Brett told you I'm going to talk about, which is bacteria. And I really only have a couple of goals for this seminar. So the first thing I want to accomplish is I want to try to convince you that bacteria can talk to each other. And if I manage to do that, my second goal is to try to convince you they're multilingual. And then the sort of third thing that I hope you get by the end of tonight is that you actually think learning about how bacteria communicate is useful, because maybe there's going to be new applications in medicine and industry. And so that's where we're going to go in the next little while. And so just to make sure that we're all on the same page when we get started, I want to just introduce you to bacteria. So my first slide shows a cartoon of a sort of regular bacteria. This would be like an E. coli cell that would live in your gut. And all bacteria look pretty much alike. They are single-celled organisms, and they're covered in a membrane, which is like their skin. So that keeps the outside out and the inside in. And then inside the bacteria are all these biomolecules that are roiling around and keeping them alive. And what's special about bacteria is that they have one piece of DNA. So they have one chromosome that has all of the genetic information that gives them their physical traits and also all of their behaviors. And bacteria are the most ancient organisms on Earth. We know that they've been here since shortly after oxygen came around, so for about 4 billion years. And so they're really primitive looking. They weren't thought to do very many tricks. Okay, thank you. They weren't thought to do very many tricks. They've always been considered to be these sort of asocial, reclusive, sort of boring organisms. Because if you watch a bacterium under a microscope, what you'll see is that it will consume nutrients from its environment, it will grow to twice its size, it will double all of these components, and then it will cut itself in half. And one bacteria will become 2, and 2 will become 4, and so on and so forth. And so since they sort of lived this primitive lifestyle, it was always thought, for the 400 years that bacteria have been known about, it was always thought that they were asocial. And what I mean by that is that when the cells divided in half, the sister cells had no knowledge, if you will, of their siblings. They simply went out and did their own thing. And so that's how we thought about bacteria until the last 20 or so years. And I want to try to show you tonight that, in fact, they have social behaviors. But in order to get you to come along on this ride with me, I want to try to motivate why you might want to learn about bacteria and the world that they occupy with you. And so this next slide is going to give you a couple of metrics about how I, or in my lab, view life on this earth. And so this person that you're looking at on this slide that's made of all these little circles, this is supposed to be a human being. And what we've done is we've done a calculation, and these circles represents all of the cells that are in the human body. So all of the human cells that make a human being. It takes a couple of trillion cells to make a human being. So that's how many cells you have in your body that give you your form and give you all of these functions that we carry out. But after a few months of life on Earth, every human being is living in a consortium with 100 times more bacterial cells than human cells. So this is how many bacterial cells would be on you when only this many, the inside of the man, are the human cells. So if you think about this metric, in fact, you're only 90% human. You're only 90% bacterial, you're 10% human. But of course, we know a lot more than about the cells. It's the 21st century, and so we have the human genome. And so I've remade the little man with the A's, T's, G's, and C's that make up your DNA, this famous double-stranded helix that has all of our genes on it. And so now the human genome has been sequenced, and so we know how many bits of information it takes to make a human being. You have about 20 or 30,000 genes. So every moment in your life, you actually have 100 times more bacterial genes that are in you or on you that these bacterial cells provide you. And it turns out that those genes that the bacteria provide are essential for life on Earth. So what we now understand is that our human genomes don't encode all of the enzymes that we need to stay alive. And so in order to have that, these bacteria are providing us all kinds of features that aren't encoded in higher organisms' genomes. And we now understand that there would be no life on Earth unless higher organisms lived in consortia with these bacteria that give us many of our traits. So you can pick whichever one of these metrics you like. At best, you're 10% human. The truth is, if we really go with the information, you're about 1% human. The rest of you is bacterial. And so hopefully you believe these bacteria live in and with you and that they give you all of these features features that actually keep you healthy. That's the sort of new thing that we're learning about bacteria. So now when scientists think about bacteria, we kind of know them as having two kinds of personality. So on the top of my slides, I have a couple of notorious pathogens that are just pictures of bacteria that were taken under a microscope. And so for the first few hundreds of years that we've known about bacteria, they've always been thought of to do these negative things. Like if these particular bacteria get in you or on you, they can either make you really sick or they can kill you. And so that's the kind of press bacteria we're getting for the first few hundred years that they've been studied. But now what we're learning increasingly is that in fact we need this consortia of bacteria living with us to provide us these traits that our own genomes don't do. So for example, they digest your food. When you eat plant food, when you eat a salad, you don't have any of the enzymes to give you access to those nutrients. The bacteria do that for you. They provide us with many of our micronutrients and vitamins, and they also, this living in and on us, all of these trillions of bacterial cells, what they do is that they actually protect you from invading microbes. So it's very clear now that these beneficial bacteria play a role hand in hand with your human-encoded immune system to keep you healthy from microbes that have no business being in you or on you. And so now what we're getting in the 21st century when we think about bacteria is that they kind of have these two personalities, the deadly ones, but then also these beneficial ones. And what my lab would like to understand, whether one is thinking about the bad bacteria or the beneficial bacteria, what we want to understand is how can it be that bacteria get any bang for their buck? I mean, they are puny. They are tiny. You need a microscope to see them. And if it was really true that when these bacteria divide, they had no knowledge of their sister cells, how can it be that they can either have us on our knees when they're harmful or, in fact, give us life itself? So what we wondered is, couldn't there be another way for bacteria to live in order for them to be able to carry out all of these tasks that we now know they can do? So how is it that they do that? And so I want to take you on a little tour of how we got to the idea that these bacteria talk to each other with chemicals and they act in groups, and that's what gives them their fantastic power on this earth. And I wanted to try to tell you this story in the way that it got discovered, because I'm trying to show you what it's like to be a scientist. So I'm just going to give you our tour into this idea that bacteria can act as groups. And it actually came from a sort of strange, anomalous observation that had to do with this squid. So this is the Hawaiian bobtail squid, and this squid lives just off the coast of Hawaii, and it lives in a beautiful one-to-one symbiosis with a bioluminescent bacterium named Vibrio fischeri. So the squid and this Vibrio bacterium live together all of the time, and the squid needs the light that the bacterium makes. And so now I want to tell you about this symbiosis, and first I'm going to tell you about it from the squid's point of view. So this squid we put in an aquarium, and we have the lights on, and so it looks kind of spastic. It's swimming around, it looks nervous, and that's because the animal is nocturnal. So during the day, the animal buries itself in the sand and sleeps. So now we've taken the same squid and we've given it some sediment, so these pebbles, and so it thinks it's morning. It thinks the sun has come up and it needs to hide from predators, so it doesn't want to be out during the day. So you're going to see it in a second. It'll camouflage itself, and now it's trying to escape from predators that would see it during the daytime and eat it. It's thorough. You only get eaten by a predator once. Okay, so it's done burying itself in the sand. It retracts those tentacles, and then I'm going to show you the entire aquarium. And if you saw this, you wouldn't know there was a squid in there. Well, there's actually about 10 squid in that tank. So this is a fantastic anti-predation device for the day. So the squid just lives in this sort of shallow, knee-deep water. It's got to hide from predators during the day, so it buries itself in the sand, and it's fine. The problem for the squid is when it comes out at night, because it can't just stay there forever. At night, it has to come out and hunt. And so on bright nights, when there's lots of starlight or moonlight, since the squid just lives in this shallow, knee-deep water, if starlight or moonlight shone on the squid and it was swimming around, the squid would make a shadow, and it would hit the sea floor, and predators that could see the shadow of the squid could calculate its trajectory, find that squid, and eat it. So this is where the bioluminescent bacterium, Vibrio fischeri, and this symbiosis comes in. So now what I've done is I've turned the squid on its back, and I hope what you can see are these glowing lobes. These are a specialized light organ that the squid has evolved that houses this bioluminescent bacterium named Vibrio fischeri. So this bacterium lives jam-packed in this light organ, and the bacterium produces bioluminescence. So this blue light that you see on this slide. And so what happens is when the squid emerges from the sand at night and it's swimming around, it has detectors on its back, so it can sense how much starlight or moonlight is hitting its back. And then it opens and closes a shutter. It's just the squid's ink sac that's underneath its body that modulates how much light comes out that's made by the bacterium. And so what the squid does is it senses how much light hits its back, and then it opens and closes the shutter, so the amount of light coming out of the bottom made by the bacterium exactly matches how much light hits the squid's back. So when the squid swims around, it doesn't make a shadow because it counter-illuminates itself with the light that the bacteria make. And so this is how it uses the bacteria for the nighttime anti-predation device. So in the day, it's invisible because it's asleep in the sand, and at night, it's invisible because it's cloaking itself with this counter-illumination anti-predation device. And so this is a fantastic lifestyle between the squid and the bacteria. But what is also amazing about the bacterium's point of view now is that the bacteria only make light at night. And so how can the bacteria know to only make light just when the squid needs it? So what the squid is doing is when it goes to sleep in the sand, it's got these bacteria jam-packed in there, and what it does is it has a pump that it's attached to its circadian rhythm. So when it goes to sleep in the sand, it pumps out like 95% of the bacteria. So now the bacteria are dilute. So even if they were making light, it wouldn't be very much. But it turns out that during the day, the bacteria grow and grow and grow and double. But then at night, exactly when the squid needs it, the light comes on. So how do the squid understand, or how does the bacteria understand when they've grown to that number? And the way that they do it is because they communicate. So what we now understand, right, in this squid, the bacteria can be dilute in the morning and at high cell density at night. And the squid is modulating that. But the way the bacteria know how to know that that is happening is as follows. So this slide is supposed to be a cartoon. This is supposed to be my Vibrio fischeri bacterial cell. So in the morning, it's at low cell density, right? The squid has pumped out most of the bacteria. It's at low cell density, so that means don't make light. So the bacteria have light turned off, but what they do is that they make and release small molecules that we call autoinducers. And you can think of these like hormones. So when the bacteria are at low density, they produce these autoinducers, these hormones, and those molecules just freely diffuse away from the bacteria, so the bacteria can't detect them. And so that says, don't make light. But then, as the bacteria grow and divide, remember they double the way I showed you in the first slide, since all of the bacteria are producing a share of this autoinducer molecule, the extracellular concentration of that molecule increases in proportion to cell number. So the more cells there are, the more of this extracellular hormone there is. And when that molecule hits a particular threshold concentration, the bacteria can detect it because it's built up to this threshold level. And what they infer when they detect that that molecule is around is they infer that they have neighbors. And then in unison, in response to detecting the hormone, all of the bacteria in synchrony turn on light. So what happens is the bacteria understand when they're alone or when they're in a community simply by whether or not this extracellular chemical, this chemical word, is around them. And then one more time to reiterate on this story, the squid is modulating this. In the morning, the bacteria are in this low cell density mode. They grow during the day. They detect that chemical. They all turn on light right when the squid comes out at night. And then the squid squirts out most of them, and they go back to this low cell density mode. And this happens day and night, so that the bacteria make light exactly when the squid needs it. And so for us, what was amazing about this was it was the first instance where scientists saw that bacteria could do something as a group. Right? And so it started to give us the hint that perhaps bacteria did have social behaviors. Perhaps they weren't quite as, sort of reclusive as we thought they were originally. And so this original observation was made by Woody Hastings in the 70s. And then the next scientist in the game was a man named Mike Silverman, who wanted to bring the tools of genetics to this story and ask, how does this circuit, this making this chemical and responding to it, work? And so he cloned the initial genes that underpinned this communication circuit. So this, again, this oval is supposed to be my bacterial cells. What was shown is that there was an enzyme that was a protein, an enzyme, that made this autoinducer, this hormone molecule. And then that molecule freely diffused outside of the bacterial cells. So the more cells there were, the more of this molecule there is on the outside. And then what got found was that there was a receptor, a partner receptor. And so this is like the hormone receptors that are on the surface of your cells that detect chemicals in your body. And so what happens is that the bacteria make this autoinducer, this hormone molecule. It builds up in proportion to cell number. There's a partner receptor on the cell surface that works like a lock-in key. It has a slot in the receptor protein where this autoinducer hormone can slot in. And when that event happens, when the autoinducer slots into the receptor, information comes into the cell to tell the bacteria to turn on the genes that make the enzymes that make light. And so it's a very simple circuit. The more bacteria there are, the more of this hormone there is at a particular concentration, which is proportional to cell number. Information goes into all of the cells, and then together they turn on light. And so this was this sort of amazing discovery by Mike Silverman about how these bacteria could do this cell communication process. And then what happened after that is that hundreds and hundreds of examples of these kinds of communication circuits got found in the bacterial world. So this was shown to not be some sort of ridiculous anomaly of a glow-in-the-dark bacteria that nobody ever heard of. In fact, all kinds of clinically relevant, important, medically or industrially related bacteria were shown to have these communication circuits. And so now what we get is that perhaps all bacteria have to have the capacity to understand when they're alone or to understand when they're in a community in order to carry out collective behaviors. So again, we now have hundreds of examples of bacteria that make these hormone autoinducers. There's always a partner protein that detects the autoinducer, and then together these bacteria turns on all kinds of collective behaviors that are not successful if bacteria do them in isolation. And so now we have a fancy name for this process. We call it quorum sensing. The bacteria vote with this chemical votes, the vote gets counted, and then they all go along with this democratic decision-making process. And in every species that have been studied, there are typically between 200 and 600 genes that are controlled by these quorum sensing cascades. So what you need to know is that bacteria typically have a few thousand genes. So something like 20 or 25% of their genes can be controlled by these quorum sensing circuits. So it's a big deal when a bacterium goes from acting alone to acting as part of a group, and they have to completely change the genetic programs that they're carrying out in order to successfully act as part of these communities. And so what is going to be important as I go on is that in many of the bacteria that have been studied, the kinds of traits that are controlled by quorum sensing are pathogenesis, right? So the idea is when a bacterium is going to infect a host, if one or a couple bacteria dribble out a few molecules of toxin, nothing is going to happen. But if they wait and they count themselves and they recognize when they have the right number that if all of them launch these programs together, they will be successful in overcoming the host's immune system. So we have many, many cases of bacteria that need these quorum sensing systems to be pathogens. So if you make mutants that can't talk or can't hear, they're completely avirulent. And so that is where this is going along in the medical lines. So now we get the idea that bacteria have community behaviors, and then we now call this entire field quorum sensing. So this is how it works, and we also know a lot about what these hormones, these words are. So the first one that was discovered was in Vibrio fischeri. So on my slides, in my cartoons, I show these chemicals as triangles, but this slide shows you the actual structure of the molecule that is the autoinducer that Vibrio fischeri uses to talk with. And so of course it was the first one that was discovered because it was the first quorum sensing bacteria known, but then many other molecules have been defined over the years. And so what I've done on this slide is to just put a smattering of a few quorum sensing producing bacteria, and then the chemical word that each one of these bacteria uses to communicate. And I hope what you can see on the slide is that the left-hand part, this ring structure, is identical in every single one of these bacteria. But these right-hand parts of the molecules are a little bit different. These are just carbons. And what you can see is each one of these is slightly different than every other one of the hormones. And so what we understand about these molecules is that there is exquisite species specificity. And so what I mean by that is if we take this Vibrio fischeri molecule and we put it on this bacterium Vibrio harvii, nothing happens. And likewise, the Vibrio harvii molecule, Vibrio fischeri, is impervious to it. So there seems to be no crosstalk in these systems. So each of these little changes on the right-hand side of these molecules confers species specificities to these languages. So what we understand now about these molecules are these are for intra-species communication. These are private, secret conversations that bacteria have with their siblings, and they allow them to talk among their own kin, and then to carry out collective behaviors. So those are the molecules, and then again, to get back to a point I was making a minute ago, we also know that this is a lot more, it's about a lot more than bioluminescence, right? So I told you in detail how quorum sensing works to control the density-dependent production of bioluminescence in this symbiont Vibrio fischeri. So I'll just tell you a little bit more about a couple other bacteria. So this bacterium called Pseudomonas aeruginosa, this is an opportunistic pathogen. This is a bacterium that just lives in the soil. You and I encounter it all the time. It's harmless unless you have cystic fibrosis, you have burns, or you're immune compromised, right? So it takes advantage of people that have these problems. And so this bacterium, Pseudomonas aeruginosa, as I was sort of telling you, to be a pathogen, the bacteria have to be able to do two things. They have to be able to make what's called a biofilm, which is how bacteria sit on surfaces. So for example, in people who have cystic fibrosis, right, they have a genetic mutation in their genome that makes it so they can't clear their lungs. You and I breathe in all kinds of gunk, and we have mechanisms to keep our lungs sterile. People who have cystic fibrosis can't do that, so they always have a polymicrobial infection in their lungs. And what happens is typically when a person is in his or her teens, they become permanently colonized by Pseudomonas aeruginosa. We don't understand why, but it's the Pseudomonas infection and the damage that Pseudomonas causes that kills people that have CF. And the reason is because Pseudomonas has quorum sensing. It sits on the person's lung tissue in this community that's called a biofilm, so a film. It sits in a community. It covers itself with this matrix that makes it impervious to antibiotics, and then so it takes quorum sensing to make that community and to build that structure, and then simultaneously the bacteria all release about 50 or 60 toxins and proteases and things that damage the person's lung tissue. And so this is a devastating, horrible disease. But if you think about it from the bacterium's point of view, this is a fantastic strategy. Again, our immune systems evolved to see invading bacteria and get rid of them. The last thing an invading bacterium wants to do is to start making these toxins and proteins that the immune system, that will alert the immune system before the bacteria can win, if you will. So what they do is that they wait, they count themselves, and then they all make these biofilms and launch and release these virulence factors in synchrony, and it's just a fantastic strategy from their side in order to make a successful infection. And so now we have, I'm using Pseudomonas as the example, but we have dozens and dozens of cases of clinical bacteria that use this trick, which is they release virulence factors as a team. And so, so that's one example. Another one I put here, this is Erwinia, and this is a bacterium you have encountered. You know how when you leave your lettuce in the refrigerator for too long and it gets all soupy? That's this bacterium. So it's a plant pathogen, and it's trying to do the same thing that Pseudomonas is doing, except in a plant. It wants to make a wound in a plant. It wants to get in there and gobble up all the goodies, right? And so what it does is it waits, it counts itself, it launches its virulence factors, and it makes a wound in the plant. But the really insidious thing that Erwinia also does is that simultaneously to releasing these virulence factors, it also releases a whole host of antibiotics that it has immunity to. And so it kills off all of the competitors that would like, bacteria, that would like to come in and get the goodies in the wound that it's made. So it keeps the wound for it and its siblings, and it kills off anybody who's trying to take advantage. Right? So this list goes on and on and on, but I hope what you can think about is that the kinds of traits that are controlled by quorum sensing are what are called public goods. They are things that bacteria give away to the world, right? They go outside of the bacteria. And so if you're a bacterium and you're living in this big world, but you're really small, once you release these products outside, you never get your own back. But if you do it through quorum sensing, so that we all release these factors together, I get the benefit of my colleague who is out there releasing these factors at the same time. So quorum sensing makes these public good production traits successful in the world. And so we have, again, many, many examples, but I think that's the way that you can think about it, is that a bacterium never gets its own product back. It depends on the collective doing things in synchrony to make these kinds of behaviors successful. And so I want to just stress that this is mostly about virulence, right, and pathogenicity, but again, we're learning now that all these good microbes live in us and on us. And so the idea that's now coming up in the field is it's not just the bad things that bacteria do with quorum sensing, all of these beneficial processes that bacteria do inside of us that keep us alive, making these nutrients, making your vitamins. Again, we're starting to understand that those kinds of traits are also controlled by quorum sensing. It's just that we haven't known about the beneficial bacteria for as long. So the focus has been on virulence, but now we're starting to learn that quorum sensing is playing a big role in how bacteria also do beneficial things, both in plants, animals, and in humans. So that's quorum sensing, and once we got this far and we started to understand some of the rules of how this works, my lab wanted to understand, could this be more complicated? So it's really wonderful if you're Vibrio fischeri and you live in this pristine one-to-one relationship with this squid. But that is not how most bacteria live. Most bacteria live in the Wild West. And what I mean by that is that they live in consortia with all kinds of species. They hardly ever have scenarios like the Vibrio fischeri one where they live only with their brothers and sisters. And so, for example, this is a picture of human skin. This is a biofilm. These are these kinds of films that I was telling you about. Your skin is covered in a bacterial biofilm all the time. It protects you from harmful things in the environment. But what you need to understand about the slide I'm showing is every one of these different shapes and sizes of bacteria is a different species. And so what we started to wonder is if bacteria are really communicating and they're really carrying out these group behaviors and these molecules are so exquisitely specific, how do bacteria deal with this kind of situation when they're in these mixed species consortia? How do they count when there's more than their siblings around? So we wanted to understand if quorum sensing couldn't be more complicated than we were thinking about in the initial parts of our investigations. So to be able to do an experiment about that, what my lab decided to do was to work on a cousin of Vibrio fischeri, which is called Vibrio harvii. This is a bioluminescent marine bacterium, but it lives free-living in the environment, and it lives in consortia with hundreds of other species. So unlike Vibrio fischeri, that has this squid host, Vibrio harvii lives in mixes. And so the reason that we wanted to use this goes back to this bioluminescence business. So what I'm showing you on this slide is just a flask and some Petri plates of Vibrio harvii. And all we did to take this picture is we simply turned the lights off in the room, and this is what we see. We're not doing anything to the cells. They're simply making this bioluminescence. And you can see it with your eyes, or you can take a picture of it. So we took the picture with the light the cells were making. And what is so fantastic about bioluminescence, right, is you can see it with your eyes. So remember, bacteria are invisible. So how do you see, as a scientist, whether they're doing something alone or doing something as a group? What was wonderful about Vibrio fischeri and then later Vibrio harvii is we could just see them all turn on light together. Right? And so we could tell that Vibrio harvii had quorum sensing because it only made light when it reached high cell density in the test tube. So we knew it had quorum sensing. And then what was particularly wonderful is that I'm a geneticist in my lab, and so what we thought we could do is to simply make mutants in Vibrio harvii, turn the lights off in the room, and look for the guys that were dark. And by doing that, we could find the bacteria and the genes that controlled bioluminescence and quorum sensing. So we used this same trick of monitoring bioluminescence in order to find how this process was controlled. And so what we found was that indeed there was a quorum sensing circuit. There had to be. The bacterium only turned on light at high cell number. So we found an enzyme that made an autoinducer, and I actually showed it to you on a couple slides ago. So it made one of those autoinducers, and then, as predicted, there was a partner protein that detected the autoinducer and sent information into the cell. But what was interesting to us is that when we made mutants that didn't have what I'll call system 1, those bacteria still only turned on light at high cell density. So that meant there had to be a second quorum sensing system. And so we went on to hunt, and we found the genes for that circuit, and sure enough, there was a second enzyme that made a second signal molecule, a second autoinducer. It had its own partner protein, and all of that information from both systems fed into the cells to tell the bacteria when to turn on light at high cell number. Right? So there were 2 quorum sensing systems. And so once we found that, then we wondered, well, why does it have 2? Why are having 2 quorum sensing systems better than 1? And what we thought is if these molecules didn't encode different pieces of information, the bacteria don't get anything extra for having 2 systems. So we were particularly interested in this new system, and so we had the genes, and so what we could do is just go look in other bacteria and ask, do they have this system? Right? So just the same way we have the human genome, we have thousands of bacterial genomes. So you can take your favorite genes, plug them in, and ask if other bacteria have those genes. And so we took the gene for the enzyme that makes the second molecule. We named that gene Lux S, for Luxor, the god of light, S for the synthase that makes the signal. And we plugged that gene into the bacterial genome database, and what we found was that unlike these systems that are species specific, every bacterium that its genome had been sequenced had this system. And so that's shown on this next slide. This is just sort of a few examples of different species of bacteria that all have a highly conserved Lux-S enzyme. And then we checked every single one of these to ask, does it make that molecule and put it out into the outside? And sure enough, every one of these bacteria made this second autoinducer, which I will call autoinducer 2. And so now what we thought was we had this sort of generic system, this system that seemed to be widespread in the bacterial world. And so we wondered, well, what are all of these bacteria doing with Lux-S and this second molecule? They're obviously not turning on bioluminescence. You know, this is bubonic plague, this is staph, strep, cholera, you know, bad breath, gangrene. It was not going to be about bioluminescence, right? So we and other scientists went hunting for the genes that were controlled by autoinducer 2 in all of these species. And on this next slide, I again just put a little smattering of different species of bacteria that have been studied and the genes that are controlled by autoinducer 2. And what you're supposed to notice is that in every case that's been studied, it is biofilm formation and virulence factor expression, right? These 2 critical steps that happen during pathogenicity. The bacteria sit on the surface as a community, and then they launch their virulence attack. And then again, I want to remind you that the beneficial bacteria have not been studied, so it's all about virulence, but the guess is that beneficial traits in sort of commensal bacteria that live in us and on us will also be controlled by this molecule. So then we wondered, well, what is autoinducer 2? And so we purified the molecule and we solved the structure of it, and it is this molecule. It is a 5-carbon molecule that's decorated by all of these oxygens. And what you need to understand that's different about this molecule than the first intraspecies molecule that I told you about is that every bacterium makes exactly the same molecule. So Lux-S does one thing. It makes this 5-carbon molecule, no matter what bacterium it's in. And so now we understand what this molecule is about. It is about interspecies communication, right? In this case, all of the bacteria can talk across species because they have a common language, which is this autoinducer 2 molecule. So we think of this molecule as sort of the trade language, or the bacterial Esperanto. That's the universal language that lets bacteria count and talk across their species. So what we now think is that bacteria must be multilingual. They are at least bilingual, and I will not be out of a job when I go home next week. There are many more molecules to find. But we think that bacteria have to be set up at least the way I'm showing you here, which is they have one language, the first molecule, the AI1, autoinducer 1 language, which is species-specific, to count their kin. Then they use this new molecule that we call autoinducer 2 to actually take a census of other species in the environment. And so we think that the computation that bacteria do is as follows. We think the sort of first easy question they have to ask is, am I alone or am I in a group? And so what they do with their receptors is they scan the environment for molecules. And that starts to tell them something about cell number. And they begin to turn on and off genes. Remember, there's usually 600 genes that are going to turn on and off at the bottom of these cascades. So first they just say alone or in a group, and they begin to carry out a program. And then the more sophisticated computation, which we actually recently showed that they do, is they measure the ratio of these molecules. So what we think they're doing is they're asking, are me and my siblings in the majority, or is somebody else in the majority in any given mixture? And then what happens is that they tailor what genes they turn on and off based on whether they and their family members are winning or somebody else is. So we think that they can distinguish self from other by using these 2 molecules, and then they carry out appropriate behaviors based on the ratio of sort of friend or foe that are in the environment. And so that is quorum sensing. And again, I want to just state that we don't think we're done. We think there will be other molecules, like ones that say who the other guy is, and my field is hunting very hard now for those molecules. But now you guys get that bacteria have these chemical discourses. You get that they carry out pathogenesis with these. And so this, what I wanted to finish this talk with is to tell you about some of the practical things that are coming out of this basic science that started with these glow-in-the-dark bacteria, but then moved into these pathogens. And so now that we, and I mean the humans, the scientists, get that bacteria can communicate, the question is whether we can do something about it. So can we make them communicate on demand when we want them to, or can we stop them from communicating on demand when they're doing something harmful? And so that is where the field is moving, and I thought I would just tell you our most brand new story in that arena. And so there's sort of 2 strategies. One is if we wanted to stop a particular species from talking, like you have an infection and we want with Pseudomonas and we want to stop that bacterium from talking, could we take advantage of the slight differences in those intra-species communication molecules and make a species-specific inhibitor, right, an anti-quorum sensing molecule? The other possibility is sometimes you want all bacteria to, your physician, for example, doesn't know what you have and wants to treat you with a broad spectrum antibiotic or a broad spectrum medicine. So the question is whether or not we could then maybe interfere with this autoinducer 2 system and make an anti-quorum sensing molecule that could shut down communication broadly. And that might be useful both in medicine and in industry. And so we are trying both of those strategies. The logic for doing the experiments is the same. And so I'm actually going to tell you a brand new story about how we're trying to interfere with this intra-species communication system in order to make a medicine. And so the idea, right, is unlike all antibiotics that you've ever taken, which kill bacteria or slow their growth, we're not going to try to do that. We're going to try to do behavior modification, right? Can we just make them mute, right, or make them deaf? And is that enough to shut down an infection? So if they can't talk, can we trick them into not being able to launch these cascades that we know cause disease? So there's a reason I've been talking about Pseudomonas for this throughout this talk, is because this is the one that we're going to test this strategy on. So I've already told you that Pseudomonas is this terrible pathogen for people that have burns, for people that are immune compromised, for people that have CF. But it is also notorious because it loves to make biofilms. It sticks to surfaces, and so it's sort of spreading around in hospitals. So this is the #1 cause of hospital-acquired infections. It's there because sick people are there, and so now it's kind of like, you go to the hospital to get your gallstones out, and you get a bacterial infection. And typically, it is from Pseudomonas aeruginosa. So it's a real problem in hospitals because it sticks to surfaces. It causes these infections when you get intubated or you get a catheter, right? And then plus it causes its own infections in CF and immune-compromised people. So it's a big, big worry in the developed world, and so we wondered, could we, sort of put our money where our mouth is, pick a pathogen that's really terrible, and try to work out these anti-quorum sensing strategies. And so again, just to remind you, we know, and this is work from many, many labs, that Pseudomonas only turns on biofilm production, so sitting on the surface, and then the group-wide expression of virulence in response to these quorum-sensing autoinducers. And we know what those autoinducers are. These are the intra-species molecules. Pseudomonas makes 2 of them. Right, so just to remind you, you've already seen this a few slides. It's a ring-like molecule with these carbon chains. Pseudomonas has one that has 4 carbons, which I'm showing you here, and then it has a second molecule that has a slightly longer tail. So what we did was we screened a chemical library for molecules that would shut down quorum sensing in Pseudomonas, and this is a picture of our best inhibitor. And what you can see, I hope, is that see this part of it, the left part of the molecule with the S there? It looks a lot like the real autoinducer, right? So this is an inhibitor because it slots into the receptor, because it looks like the autoinducer. It slots into the receptor, it jams the receptor, and it doesn't allow the real autoinducer to get in there. And so it shuts down quorum sensing simply by competing with the bona fide signal. Okay, so that's our molecule that we got. It's a synthetic molecule that was made by a chemist. And so now I want to show you that, in fact, you can shut down quorum sensing in Pseudomonas. So I hope you can see this slide. So remember, the first thing when Pseudomonas needs to make an infection, it sits on a surface and then it spreads out in this biofilm structure. And so what you're seeing here, this is a Petri plate, and what we've done is we've just put a little drop of Pseudomonas right in the middle of the Petri plate. And then you see all of these arms coming out from the middle. That is Pseudomonas swarming to the edges of the Petri plate in this community that we call a biofilm. So this is what Pseudomonas normally does. If we put the inhibitor in the Petri plate, so we just add that molecule that I showed you, and then we put our drop of Pseudomonas right there, what you see is that Pseudomonas can't swarm, it can't make a biofilm. So that was good news for us. That was the beginning of showing that perhaps there's merit to this idea of shutting down quorum sensing and stopping these virulence traits that we know are important. So that's the biofilms, but then remember, I also told you that Pseudomonas secretes all of these toxins, these virulence factors. A really deadly one of these virulence factors is a molecule called pyocyanin. It is a molecule that kills human lung cells, and it's called pyocyanin because it's cyan, it's green. So the molecule is actually green. So when Pseudomonas secretes this molecule, the entire culture turns green. And so that's interesting for us because we can monitor it just by looking at the culture. And so now you're looking at 3 test tubes of Pseudomonas. And so this is wild-type Pseudomonas. It's at high cell density, quorum sensing has happened, and it's made this pyocyanin green virulence factor. And so what you can see is that the culture is green. If we deliberately make a mutant in Pseudomonas that doesn't have quorum sensing, right? So it doesn't make the autoinducer, it doesn't have the receptor, the culture is colorless, right? So that shows you that quorum sensing controls production of this virulence factor. If we take Pseudomonas, wild-type Pseudomonas, and we add that inhibitor, what you can see is that the culture is colorless. So now I've shown you 2 things. First, that inhibitor stops a biofilm, and second, that at least in a test tube, it stops Pseudomonas from making this important toxin pyocyanin. So that gave us some confidence that, again, that we were actually going in the right direction. So we wanted to ask, could you shut down Pseudomonas in an animal infection? And so the last experiment I'll show you is here. We have an animal model for Pseudomonas infection. And so what we do is we measure the % animals that are alive over time. And so, of course, if you don't give a Pseudomonas infection, if you do a mock infection, the animals are alive. In this case, for a day, they will be alive for a lifetime. Right? But if instead we give the animal a Pseudomonas infection, what you can see is that all of the animals die in a day. So if we do, that's because this is a deadly pathogen. And so if we do that experiment, but simultaneously we give our anti-quorum sensing inhibitor, what you can see is that now the animals live. Right? So this now is really beginning to show us that, in fact, this idea of thinking about how to treat infections differently, instead of killing bacteria, simply stopping them from talking and letting the immune system get rid of them, that maybe, in fact, this could be a potential way forward to think about novel kinds of antimicrobials. And I want to be really clear right now, this is not a medicine. The molecule that I showed you a couple slides ago does not look particularly medicine-like. It isn't safe, it's toxic, it's not particularly potent. You have to take a really big dose to get this to work. But it did give us the confidence that, in fact, we should begin to do the kind of medicinal chemistry that is required. You can build in potency, and you can build in medicine-like properties so that the molecule goes where we want it to go and stays there as long as we want it to stay. And so now we are actually actively working hard on improving that molecule to make it resemble or hopefully become a medicine. But that's the place where we are right now in our studies. So the steps in the Pseudomonas work and with other pathogens, right, is to actually make these lead molecules, what I'll call lead molecules, look medicine-like and be safe. That's one thing. The second thing is, remember, that at least for Pseudomonas, that it makes these biofilms, right? It gets on these surfaces like of intubation tubes and catheters and things like that. And so what we're also trying to do, we've teamed up with the engineers, and we're trying to embed these anti-quorum sensing molecules into materials. So we'd like to make biologically inspired materials, and maybe you could think about infection-resistant materials or implants or stents or knees or breathing tubes. And so we're working really hard to actually think about those kinds of more industrial and medical applications. And then, of course, remember, there's many other pathogens. This is just our proof of principle experiment, and there's a number of different molecules to work on as well. So that's the applied part. And then to truly finish this talk, I want to go back to the very basic science and how we got where we are today. And I hope what your takeaways are the following. I hope you think that bacteria can talk to each other. They don't have voices like we do, so we think that their primary mode of communication is through chemical words. What quorum sensing gives bacteria is the ability to be multicellular. And so as scientists, we think about evolution a lot. Bacteria were the first organisms on this earth. We think and we come from bacteria. We evolved from bacteria. So we think that bacteria in these ancient organisms, the principles for multicellularity, for how group behaviors can be robust, for where leaders and followers and cheaters come from, we think all of that is embedded in these basic bacterial systems. And so that if we study bacteria, like these bioluminescent ones, we'll be able to understand at least the beginnings of the rules for how multicellularity happened on this earth and why it became so successful. What are the features of multicellularity that are better than being a single cell? And we think quorum sensing is a part of that. And then again, in a statement I already made tonight, I hope what you believe is with these two molecules, one that says you're my twin and one that says you're other, we believe that bacteria can distinguish self from other. And again, I would argue that that's exactly what happens in your body. It's not like your kidney cells get all confused with your heart cells every morning. And that's because these different hormones, these different chemicals in your body tell different cells that are located in different places to do different jobs. And again, we think that that exists in this very ancient primitive world of the bacteria, and that perhaps by studying quorum sensing and these different molecules and what information they encode, we can help colleagues who study higher organisms to understand how the human body works, both in health and in sickness. And then again, to get back to the sort of practical part, we think that there's a very applied part of our work, even though it came from this beautiful glow-in-the-dark bacteria. In these last couple of decades, it has absolutely led to a
cottage industry of scientists trying to make anti-quorum sensing molecules to stop harmful bacteria from doing harm. And then now the sort of name of the game is to actually make pro-quorum sensing molecules. We use bacteria to make us things. They're gonna make our biofuels. They make us all kinds of medicines. We need them to grow plants reliably, right? If we could find out enough about quorum sensing in these beneficial bacteria, maybe we could make pro-quorum sensing molecules that would help bacteria, again, in medicine and in industry and in agricultural, right, to do these things and solve sort of pressing world problems by making these bacteria act like little collective machines. And then finally, I should finish with a confession, which is that this business of either thwarting quorum sensing or improving quorum sensing, the bacteria figured this all out billions of years ago. So we, went, aha, maybe we can interfere with quorum sensing, but lots of my colleagues are going out and looking in natural environments, and sure enough, you'll find some quorum sensing bacteria, and the guy next to it in the dirt eats its autoinducer, or it makes an inhibitor, or it makes an enzyme that cuts the autoinducer in half. And so these poor guys are out there trying to talk, and all these shenanigans are going on in these competitive environments where they're actually naturally shutting one another down in order to, thwart quorum sensing. And so maybe we should stop trying to do this in the lab, and the best strategy for going forward is to go out and ask, what did the bacteria evolve? Because the strategies that are out in the natural world have won over 4 billion years, and the idea that Bonnie Bassler, a 22-year-old chemist, can make a better molecule is ridiculous, right? And so we have a pony in that race, as do a lot of other people, and I think we're going to find these molecules, and we're also going to find just a treasure trove of new molecules that bacteria make that humans can use to do all kinds of wondrous things, both in medicine and in industry. And then my last slide, I get the pleasure, the absolute pleasure of getting to come and give this, be in this lovely venue in this wonderful theater. But of course, I didn't do most of this work, and so this is a picture of my lab. We live in Princeton, New Jersey, so right outside New York. And every experiment that I told you about tonight was done by somebody's hand and somebody's brain in that picture. And so I'm super proud of them. They are a rush to work with, right? And I just want to say, for those of you who aren't scientists, that maybe you don't get a chance to be around scientists so much, at least in North America, what drives science is this 22 to 30-year-old demographic. Whenever you hear something amazing that when you read it in the newspaper that happened in the natural world, or that some engineer made some amazing gizmo, typically it was somebody between sort of 20 and 30 years old. They are the absolute brain that drives basic science, at least in the United States, and I think over here in Canada too. And it's just a privilege to get to work with them, because they weren't thinking about bacteria the way other people thought about it. They had all these new ideas, and they sort of put their brains and their hearts and their hands to work and came up with both the understanding of quorum sensing and then these ideas that maybe we could interfere with them. And so there they are, my heroes, and I'm really proud of them. And I just want to thank one more time the Peter Wall Institute. Is an absolute honor to come to this, and I thank you, and get to give this talk, and I thank everybody for coming out tonight to listen. And I want to thank the band too. So thank you.
I'm Kelly Crow from CBC, and it's my great honor to be here and pleasure, and that is fascinating.
Thanks.
Absolutely fascinating. And so this is the way it's going to work. I'm going to just ask a couple of questions because I can, because I have the microphone and because I get first crack. But people should be lining up at the microphones and asking your own questions, and we also have questions from Twitterverse, which I'm going to try to coordinate.
Good luck.
So first of all, I wanted to say, you make that look easy. It's all so obvious when you spell it out. Well, duh. They talk to each other. But I know that that wasn't how it went. And I know that it's a lot of hours and a lot of false starts and a lot of frustrations, et cetera. So I wanted you to talk a little bit about how you tripped over this thing in the first place.
Right. So hindsight is clearer, right? I mean, that's a goofy thing to say. People say that, right? And so I think the so bacteria have been studied for 400 years. Why did this only get discovered 20 years ago, right? Given especially that we get now, it is so widespread in the bacterial world. And I think part of that was hubris, and that includes in the microbiologists. There was just this idea, the bacteria gave us really important things, DNA, RNA, proteins. We learned about those things from bacteria. Like, they would give us the parts list, but the fancy stuff would all be in higher organisms. So there was just a sort of snobbery that they sort of looked too primitive. They couldn't possibly carry out these sort of fantastic feats, right? And so the problem was the microbiologists didn't think that either. And then again, and so the way I tripped over this, right? Remember, as I tried to tell you tonight, there had already been this finding of Vibrio fischeri. So I didn't find that. It was known that bacteria turned on light at high cell number, and that there was a molecule involved. And so I had the great good fortune of, when I was a graduate student, of being at a meeting when this man, Mike Silverman, told that story. And I remember, I was like 25 years old, and I was sitting out there, and I was thinking, that guy, so he told it, these bacteria are doing this thing together, right? And what's so awesome is bioluminescence. We can just follow it. Maybe a monkey could do that. You can just see it with your eyes, right? And I remember sitting there thinking, I am smart enough to turn the lights on and off in the room and figure out which of those guys are dark, right? Number one. And number two, I thought, like, that guy either has the most amazing idea or he's crazy, right? And so but it just captivated me because it was such a new idea, even though now it seems so obvious. And so I literally ran up to the podium and I said, you have to give me a job as a postdoc, and he did take me in. And it turned out, because it was this sort of fringy system, right, a bioluminescent bacteria, it wasn't a pathogen, that when I got to his lab, there was nobody working with him, right? And so he was kind of this loner working on this sort of anomalous bacteria, and we just went to town. And and we never thought it was going to be the story I told you tonight. We thought we were going to understand something about sensory perception, something about collective behaviors, and we thought that Vibrio fischeri and Vibrio harveyi were good models, but we really didn't think it would be in other bacteria. And even then, that was like kind of goofy. And so, Kelly, I have to say that half my life I spent thinking my lab is so awesome. Look at all we found. And then the rest of my life, I think, this is all obvious. Why did it take you so long to get, of course they can talk across species, and so that's the fun and the torture of science.
But as you've been working on this, the questions you're asking have taken on new urgency. So maybe you could talk a little bit about the climate when you started, and we still had lots of antibiotics, and people could see that there would be an issue, but to where we sit right now, which is where scientists are banging their heads against the wall trying to find something new. So talk a bit about that.
New antimicrobials, right? So you guys all know there's this global problem with antibiotic resistance because of the misuse and overuse of the arsenal of antibiotics that we have now. That's part one. The other part is, again, and this is arrogance, like antibiotics are the greatest public health measure, next to clean water, that have ever happened. And scientists could not imagine right after World War II that bacteria would get resistant. And of course, that's ridiculous when you think back about it and we understand the mechanisms of resistance, just random mutations, of course. And so there's two problems. One is that there was not a lot of work in the space of making new antibiotics because it was thought that the bacterial problem was solved. So part of it is nobody was studying it hard, and it's hard. And then the second thing is that, of course, now we've run out of the antibiotics we do have, and so there is a tremendous urgency among microbiologists that we have to do something, and that there was sort of this 30-year gap where we, didn't do much. And then also, I hope what you got from my talk is this idea that maybe you can have traditional antibiotics, which slow the growth or kill bacteria, but maybe you can make behavior modification molecules. Like maybe that would be a new, cool, sort of slick way to treat bacteria. And so, of course, that is unproven, what I just showed you, because no antibiotic there's never been an antibiotic that didn't slow the growth or kill bacteria. But now there's a lot of work in the space of all kinds of behaviors. So the behavior I like is communication. Can we interfere with that conversation? But other people are trying to make bacteria that can't swim or can't secrete a particular virulence factor, right? And so there's a whole lot of encouraging work in the area of these sort of behaviors, as well as people trying to find new molecules that kill bacteria and work like what you would think of as a traditional antibiotic.
So what are the challenges ahead now with your molecule?
Yeah, well, the challenges are the challenges is that there's no path that we can follow for how do you make that molecule into a medicine, because there has never been an antibiotic like that. So that's interesting and tricky to try to navigate. And again, as I alluded to, we have to make it safe. We have to show that it works, right? I'll confess the thing that keeps me awake at night. If you listened, which you did, to the experiment I showed you, we gave the molecule when we gave the infection. What we have not shown is, can you shut down an existing infection? Right? Our molecule is not potent enough to do that, right? So if you really went to unlike in that experiment where we gave the animal the infection, right? If you were sick and you went to your physician, you have an infection. So quorum sensing has happened. And so the question is, does the conversation that bacteria are having, we know they need it to get the infection going, do they need to keep communicating during the time of the infection? So the big question for us is, can you give it after the fact, and for how long, right? But we need a more potent molecule to be able to do that experiment. Right?
So what next?
We need to make, so that molecule and so there's two things. One is we are working on that molecule to add little bells and whistles and make it look like a medicine and make it much more potent. That's part one. And then the second thing is that experiment where we got the molecule I showed you gave us huge confidence. We're like, holy cow, this could actually work. And so we are seriously screening for other molecules, other pathogens. That may not be the best starting molecule, right? And so that sort of mini experiment that was the proof of principle told me in my lab that we should actually not have one graduate student working on this, but we should actually really focus on making that a big effort in my lab. And so we are.
Okay, well, I see some people,
There's
at least I see
there.
one person over there.
Yeah. There.
Oh, one person over here. Okay. So I've been told the rules are keep the question quick so we can get more questions. And so why don't you start?
Sure. So I was wondering, you talked a lot about trying to impede communication and stuff like that, but presumably these bacteria are communicating so that they activate and do crazy things at the right time. How much have you talked about or discussed or thought about, like, just telling them to do stuff straight away, just setting up and flooding the entire system with communication molecules so they just try and do stuff early and then the immune system can spot them or something?
Do you want to have a job in my lab? Yeah, exactly. So I just want to tell you that when you come up and you're the public lecturer and you say we're going to make them hyper-virulent, it does not make you very popular. But in fact, you guys get his strategy? He's absolutely right. We're doing that. So the idea is, I showed you, if you could interfere with quorum sensing, they never launch. Your strategy, which I think is equally valid, is if we could give a really strong pro-quorum sensing molecule and the bacteria show their stuff before there's enough of them to get the benefit of the collective, the immune system will hunt them down and get rid of them. And so we are absolutely working on that. So you could imagine you could just use the autoinducers, right? Right? But in fact, they are also not medicines. They're not potent enough. We need a sort of super autoinducer. So you're right. So when we do these screens, we do two. We do one for things that shut down quorum sensing, and we do another screen for things that turn on quorum sensing. And I don't know if either of these strategies can work or which one is better. And so we are doing both. But I hope everybody will forgive. It's easier to understand, I think, that one, right? And so that's why I use it as the example. But I think that's absolutely valid. Thanks.
Can you speak just quickly, I don't know if this is beyond your area of concentration, but can you talk about possible applications for viruses?
A little bit. And so do people know, so viruses live inside of cells, right? So viruses can infect your cells, they can infect bacterial cells, right? And viruses, why they are so insidious is because they don't have very much machinery of their own. They're not really alive on their own. They get inside of a cell and they hijack your proteins, your cellular machinery, to make more of themselves. Then they bust out and they go and they do it to the next cell. So that's what a virus is. And so they can be pathogens, right? You guys know a lot of viral diseases. And so they typically have very few genes. And so the idea, and there are 2 papers on this, not from my lab, is that they won't have enough, and this could be arrogance 10 years from now, they don't have enough genes to have their own quorum sensing system, but in fact they're eavesdropping on the host. So there's a new story, that a paper that came out that shows that the bacteria the viruses are sort of waiting on, like they're waiting for quorum sensing to happen, because that means there's lots of neighbors around, and so at the peak of quorum sensing, that's when you want to bust out, because then there's going to be a cell next door for you to get in. And so what it's been shown is that the number of that when viruses break out of cells is actually controlled by the host's quorum sensing system. And so I think that, and that's what viruses do for a living. They take the host machinery and they use it for their own purposes. So the idea that they're using the internal machinery to make more of themselves, and then the quorum sensing machinery to know exactly when they should leave and go to the next cell, I think that is an awesome new idea.
So that means then that humans' own quorum sensing cells could be manipulated in order to control viruses and...
Possibly. So now humans don't have, we don't know if human cells have quorum sensing yet, but the idea is possibly, and that yes, and that if we could learn enough to do these things safely and reliably, and this, right, is that, like he asked, pro or con, eventually we should be able to do something about that, right? Like some clever young graduate student will do that. I mean, that story is now out there, about viruses tuning into the host, the bacterial host quorum sensing system. So why not do that? Something about that. Yeah, so I think that's on the horizon. Yeah.
Next question.
So in your strategy for, you're basically screening a chemical library to get an inhibitor which will compete with the autoinducer, but basically you, as we saw that there's a family of autoinducers from other species which will all basically have a conserved moiety in the molecule. So can we think of inundating the system with existing autoinducers from other distantly related species so that we don't have to look for an alternative chemical molecule or synthesize one?
Where am I? Right? So, yeah. So I am giving a talk on Thursday to the microbiology department, and that you just gave the punchline. Okay, so thanks. So the answer, that is an awesome idea. So the question, which I will now say, if you don't mind, in my own words, right? If you looked at my inhibitor, it was an autoinducer with a big bump on the end, right? When you looked at the list of autoinducers that I showed, they got bigger, 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 bigger. So it turns out you have it. Big autoinducers are inhibitors, natural inhibitors of bacteria that use small autoinducers with short tails. It works perfectly. And so we could have saved a lot of time had we done that experiment first. But when we got our inhibitor, we're like, this looks like an autoinducer that's got a long tail. And then we went back, because we had all those in our collection, and we tested exactly the experiment that you just said, and it worked perfectly. So I think that molecules that naturally exist that are competitive inhibitors, that's a good idea. And then I think you can find molecules that work by other mechanisms, not competitive, that bind elsewhere on the receptors and do other things, that those would still, you might need to get out of synthetic libraries. But again, people are mining, bacteria for naturally made molecules that are anti-quorum sensing ones, but those are the best ones. But they're not medicines, right? We still have to make them more potent. Yeah, but that's an awesome idea.
Thank you.
I'm going to go home and give someone an F. Yeah.
Can I just interrupt and take a few Twitter questions? Do you mind at the mic? Is that okay? Okay. So a question here. Do any of these quorums, as this questioner asked, have queen bees, or are they all equal within the quorum?
Right. So we don't know the answer, right? So we don't know the answer to that. the molecules, what's so interesting about the molecules that I showed you, so the same way we can put genes into genome databases, you can put molecules in and say, what's the closest molecule to that? The closest molecules to the ones I showed you are actually insect pheromones, right? So insects, ants, and bees, and cockroaches, they have all these amazing chemical worlds that they live in. And so this idea that these might have been the progenitors is there. And so the answer is they sort of resemble insect pheromones. And then we are behind the people who study ants and bees in understanding the sort of social architecture of the system. We, meaning the microbiologists, do not understand who the leaders are and who the followers are, right? But there has to be somebody that gets it going, right? And so there could be a queen, right? Or at least the leader, right? And that we don't know about yet, right?
In one of your papers, you talked about freeloaders too. There were people who weren't paying,
right.
who weren't.
So there's cheaters, right? And so they're right. So remember, these are public goods, and I give them away. They are expensive. If I were going to design this, I would make the autoinducer, and I would never turn on the 600 genes. I'd let you do it, Kelly, right? And so there are absolutely cheaters, but somehow there have to be molecular police, right? That there's a penalty for cheating. Otherwise, quorum sensing would not exist on the Earth because it wouldn't be stable over evolutionary time. If you always got cheated, you end up losing. And we see quorum sensing is there. And so now the evolutionary biologists are way in the fray to try to understand how is cheating control. And cheating means you get mutations, that let you take advantage. And so how is that controlled? And so I think that we're going to understand a lot about that in the next few years.
Another question here from someone is whether or not this is involved in bacterial death. The question is, what stops bacteria, especially healthy ones, from replicating? Other bacterial signal inducers, or a lack of siblings, or something else?
Wait, can you say that? Sorry.
Well, I think they're asking whether or not the communication affects the multiplication of bacteria.
Right. And so thanks. So far, right? And remember, so far, quorum sensing is not about growth rate, at least not in a test tube, in a lab. Obviously, if you don't have quorum sensing out in the wild, you're going to lose because you don't have the virtues or the benefits of collective behaviors. But at least in the lab, quorum sensing changes the genes that turn on and off, but it doesn't change how fast or slow you grow, right?
Okay, do you want to, we'll take that question over there.
With regard to the species in autoinducers, I'm not familiar how far they spread. Like, is that something that could be used as sort of a really specific, like a diagnostic test for finding these infections in hospitals really early on?
So maybe you guys, he was a little quiet. So he's asking me, could you actually take those autoinducers and actually use them for diagnostics, like they're a fingerprint, of a bacterium. And so there's a lot of effort in that world of applications to try to ask, could we know enough about different autoinducers that you could make a little dipstick, right? And then it would light up, it would be like a biosensor, right? And then it would say, you have bacterium X. And so people are trying to do that. Again, we only know a few of these molecules so far. It's hard to make sensors, because the molecules are so closely related in structure, it's hard to make sensors for one that aren't the other. But I think it can be done. It's early days, and there's a lot of activity in that arena. Because if you could do something faster, which is the name of the game when you're sick, how soon does your physician know what is wrong, what bacterium you have, right? It drives your treatment. And so I think that, or even just for contamination in meat packing plants, there's all kinds of ideas about actually using the molecules not for the kinds of things that I'm telling you, but as a fingerprint. And so I think that's a great idea.
I'm curious what you've noticed as you've been working on this in terms of the environment. It sounds like you're working in isolation in that lab, trying to understand the luminescence problem, and then all of a sudden, has there been this explosion? Is it competitive right now? Are people racing? You say you're all looking for particular molecules for doing particular things.
So there has been an explosion, but I think that's it's called quorum sensing, Kelly. The group gets more than the individual, right? Yeah, and so, yeah, yeah. So anyway, so there are hundreds of labs that work on this now, which is awesome because this field is in its infancy. There's so many more molecules to find. There's so many more features to find that nobody's tripping over one another. You know, like all the questions you guys are asking me, I don't know. You know, and so there's plenty to do. And then simultaneously, and this is Brett's field, the other thing that happened, sort of, these two things happened at the same time and then they merged. And this is this idea of the microbiota, which is where you have all these fantastic bacteria living in and on you that are driving your health and that are goofy when you get sick. You know, and so there was also an explosion in just learning that those bacteria are there, learning that we need to understand them, and that is going to be essential for health in the 21st century. And so that field also exploded, and they're connected, right? Because all these bacteria are talking to each other, you know, but we know so little about these beneficial bacteria and who's doing what and how they make you healthy and who gets goofy when you get sick, that both of those things happened basically over the last 2 decades. And so bacteria are having this renaissance, right? It was all thought that it was solved. And actually, you know, with me working, like, you know, like if you didn't work on neuroscience, it meant you were a dumb scientist. Like who would work on bacteria? Like weren't all the problems done? You know, and so in fact, it's just been a wonderful 20 years, you know, where there's this idea now that these little critters, you know, can solve food, health, energy, and the environment if we learn enough about these treasure troves of pathways and molecules and tricks, right? That there's just there's an urgency. It's not that we're tripping over each other. We can't do it fast enough in both of those fields, right? Because they're only 20 years old. Yeah, so it's just a wonderful, so any young people out there, you should definitely become a microbiologist because it's just an amazing time right now.
So I think we have only about 5 minutes left, so anybody who wants to rush to the microphone. I have a last question that occurs to me as you talk about this, which is that when you started out, you didn't imagine that this would lead to a potential answer to antibiotic resistance. What does it say about basic research? Because this is a big question in Canada, I know, and around the world, about whether scientists need to have a commercialization goal when they're funded, or whether they can just follow, as you put it, follow their nose. Can you talk a little bit about that?
Yeah, so of course I'm a big believer in basic science. Like, so my sort of bottom line is that scientists, you know, you work on bacteria because you want to make people healthier. Right? So in our hearts, we all are do-gooders. We want applications to happen. And there's sort of an adventure that happens from the discovery all the way to the application. And my part of that adventure is at the very beginning, like making discoveries about how things work. And my view is that people like us are essential because everybody wants applications, but the pipeline for applications come from basic science studies. And so if you don't make the discoveries, there's nothing to apply. And so I think this idea of letting scientists sort of follow their noses, be curious, you know, work on a glow-in-the-dark bacteria that had never even hurt a fish, right? That you can, you know, trust isn't the right word, but that we have in our minds applications and we have in our minds health. And so, you know, I think, you know, the advances, if you look at the, you know, we were talking about this before, the greatest advances in the 20th century in biomedicines, the applications, all of them came not from the thing the scientist was doing it for, like, you know, MRIs, statins, Viagra, you know, that might not be the greatest, but they were all scientists working on something else because they were curious, and then there was this aha moment that I could do this other thing. And I think there has to be room in the system for people like us to go on that adventure, because over and over through history, you know, it has paid off big time.
Okay, well, thanks very much. That was
Thanks.
wonderful.
Yeah. So now you know when I get an invitation to a meeting, I look at the speaker list and like there's B. Bassler before B. Finlay. I thought, uh-oh. Bonnie, that was extraordinary. That was just absolutely lovely. And I think, you know, your passion for science and telling the scientific story of how it evolved. She's completely right. I mean, we don't go and say, I'm going to go solve this. You sort of follow your nose and then complunk, you're somewhere else. So thank you again. Extraordinarily beautiful talk. I really appreciate it. Yeah. And yeah, very nice. And Kelly, lovely job. Great questions. Great handling them. I really appreciate that. And you know, as you know, Kelly is a media person. She spends her life on CBC, convincing people to watch CBC that microbes are important. And I really appreciate that. And we'll talk some more. So thank you, Kelly. I need to quickly thank our sponsors again, CBC, the TIE, CITR, and the Georgia Strait for their sponsorship. I also want to thank the staff at the Peter Wall Institute. These things don't just happen spontaneously, and so they've put this whole thing on, huge amounts of work to them, so thank you, guys. And also the Vogue Theatre and the people that work here. I mean, this is a grand old Dame theatre. What a lovely place to come and talk about quorum sensing. Thank you, Vogue Theatre. We have a quorum. Yeah. So finally, thank you, all of you, spending an evening hearing about things you probably never even thought about before, but I guarantee you that your world has changed when you walk out this door. You've learned things, and that's what science is all about. So to find more about the Institute and our lecture series, you can visit the Peter Wall Institute website at pwis.ubc.ca, and you can sign up for our newsletter. So with that, I say good night, safe travels.